So I wanted to ask you a little bit about where you see your work going in the future. So you talked about the COVID-19 research, which is obviously very relevant right now, but we just wanted to ask you a little bit about the future directions of your lab. Do you see it expanding in size, but also in the types of projects you want to do?
Absolutely. So my lab, we're hoping, we just got funding for a few other projects, and so we are expanding. Ideally, where we want to be in 5 years is a lab that's well established in using stem cells to model the lung, whether it's for modeling disease or development, or whether it's disease modeling, or whether it's for precision medicine or regeneration, whatever. What we hope to do, and this is what I call my blue sky, which is my I wish I can do it kind of project, is to eventually refine our stem cell models, because right now our stem cell models are quite simple. It's just the epithelial cells. But as I mentioned earlier in the interview, that the lung is comprised of at least 60 different cell types, and they're not all epithelial cells. So you have vascular cells, you have immune cells, a whole bunch of other cell types. So what I would love to do in the future is to create a model where we have all these cell types all integrated together to really fully recapitulate the human lung, so that we can really understand it better and provide a preclinical model for other researchers to study development, or to study a drug, for example, whether or not it's going to work before they give it to the patient. A lot of these type of studies are done in animal models, but as you can imagine, the animal is not the man, and so their physiology is going to be quite different. So having a more relevant human clinical model, I think, is going to be important in assessing the efficacy of any type of therapeutic. So we are aiming towards trying to create 3D models of the human lung through bioprinting. And so hopefully in 5 years we'll get there. So that will require us to make more of these other lung cell types that I mentioned, and then try to integrate it into this 3D structure. That's my goal. Whether or not we get there, we'll see.
That's amazing.
So thank you so much for all your answers to all of our questions. I'm sure all our viewers are going to be very happy to hear about your lab and also learn a little bit about getting involved in research. So we will be posting this interview to our social medias and our YouTube for viewers to access, just for a recording.
Yeah, and I'll also post the link. We talked a little bit about those research courses and opportunities, so I'll post the link to some of those below. And also some of your recent publications. There's some, I was reading one about the CF Canada Sick Kids program for individual cystic fibrosis therapy. And then I think a more recent publication of yours that was how to generate patient-specific lung-like progenitor cells to study lung function. So we'll link those below for our interested viewers. And yeah, thank you so much for everything.
Thank you. It was a pleasure.